Hello everyone and welcome. Today we are going to explore one of the most fundamental processes in all the biology, that is the mechanism of the enzyme action. These molecules are the reason life can function at the speed it does. Think of an enzyme as a biological shortcut. They are special protein that act as the catalysts, meaning they speed up the chemical reactions without being consumed themselves. A reaction that might take hours or even days on its own can happen in less than a second when an enzyme is involved. This incredible efficiency it what allows us to digest food, build muscles, and essentially live. Our focus today will be on specific powerful enzyme from the digestive system, that is the trypsin. All enzyme action boils down to one critical location on the enzyme structure, that is the active site. The active site is a unique pocket or groove where the enzyme's target molecule called the substrate binds. Early scientists described this interaction using the lock and key model, suggesting the enzyme, that is a lock, was rigid and only the perfect substrate, that is the key, could fit. However, a more accurate model, and the one we use today, is the induced fit model. In the induced fit model, the active site is flexible. When the substrate approaches, the enzyme actually changes its shape slightly to perfectly wrap around the molecule, like a glove fitting in a hand. This tight hug is critical because it puts strain on the substrate's chemical bond, making them much easier to break. This is the first essential step in any enzymatic reaction, that is the formation of enzyme-substrate complex. Now, let's look at the specific example, trypsin. Trypsin is an enzyme produced by the pancreas and released into the small intestine. Its function is to digest long protein chains from the food we eat. Proteins are massive, complicated structures, and our body can only absorb them after they are broken down into smaller units, peptides. Trypsin is the surgeon that makes the precise cuts. Trypsin is what we call a serine protease. This means its core mechanism involves an amino acid, that is serine, and it breaks the peptide bonds. But its most remarkable feature is its extreme specificity. It does not cut proteins randomly. Trypsin cuts a protein chain only after two specific amino acids, that is lysine or arginine. These two amino acids share one distinct feature, that is a strong positive electrical charge. Trypsin achieves this precision using a part of the active site called the specificity pocket. This pocket is deep and contains a negatively charged amino acid aspartate at the bottom. Think of it as a chemical magnet. When the protein chain enters the enzyme, the positively charged lysine or arginine is powerfully attracted and pulled deep into the negatively charged pocket. This locks the specific target bond of the protein right into the position needed for the enzyme to make the cut. The actual cutting action is performed by a tiny synchronized 3-person team within the active site known as the catalytic triad. These three amino acids are serine, histidine, and aspartate. Their job is to cooperate to make the serine molecule an incredibly powerful nucleophile. That is, a molecule that strongly seeks out a positive charge, allowing it to attack and break chemical bonds. Here is the synchronized action. First, aspartate. Aspartate acts as a stabilizer, anchoring the entire system. Second, histidine. Histidine acts as a chemical sponge, temporarily accepting hydrogen atom from the serine. By losing a hydrogen atom, serine's oxygen atom becomes highly reactive and unstable, and it's now primed for the attack. This arrangement is a perfect example of how a three-dimensional folding of a protein creates a powerful, temporary chemical environment that would not exist otherwise. Now let's walk through the two main steps of the chemical cleavage or cutting of the protein bond. The first is breaking the first bond, and the second step is resetting the enzyme. So the first step, breaking the first bond. The protein chain is locked in a place via the specificity pocket. The reactive serine oxygen attacks the carbon atom of the peptide bond right next to the lysine or arginine. This attack is so strong that it immediately breaks the peptide bond. The first half of protein chain is released. The other half of the protein remains temporarily attached to the serine. This is called the acyl enzyme intermediate. Resetting the enzyme. Step 2. The enzyme is still holding the second half of the protein. Now, a water molecule from the surrounding environment enters the active site. And now histidine comes, and histidine, the helper, steals the hydrogen atom from the water molecule, making the remaining hydroxide group highly reactive. This reactive hydroxide group attacks the bond, the second protein piece of the serine. This final attack frees the second protein piece, and the hydrogen atom is returned to the serine, which snaps back to its original neutral state. The enzyme trypsin is completely regenerated and immediately ready to begin the cycle again with a new protein substrate. The mechanism of enzyme action, exemplified by trypsin, is fundamental. It demonstrates how protein structure dictates function. The specificity pocket ensures the correct substrate is positioned. The catalytic triad provides the chemical power needed to overcome the enormous energy barrier of breaking a strong chemical bond. Without this incredible fast-paced mechanism, without these reusable molecular machines, our bodies simply could not process the energy and materials needed for life. The food would sit in our stomachs for months, and we could never generate the energy needed to move, think, or grow. Thank you so much. That is enough for today.